Hello, everyone, and thank you, Sibi, for that lovely introduction. Let's make sure that we're ready to go here. We are. As Sibi said, my name is Dr. Tovah Salcedo. I also want to introduce today a special guest to help me with the presentation. Our assistant is Dr. Novo. Look for him popping up around our slides. I would like to welcome everyone and make a quick technical announcement. Today's webinar will be conducted in English. Our automatic captions today are courtesy of Microsoft PowerPoint. However, because we are a global company, you can email us at inquiry_us@novogene.com to request support in your preferred language. This email address will be shown later, and we will provide these slides so you can easily click through. So let's get started with today's presentation. We're going to meet RNA-Seq at Novogene, all the way from samples to data. Let's start with a problem that most people understand. Childhood cancer is scary. Neuroblastoma is one type of childhood cancer. It remains poorly understood. We wonder, why is it so dangerous compared to other cancers? We also want to know, how can we study this if we don't know which genes are responsible? What if we know one or a few genes, but not what they do? One answer is eukaryotic mRNA sequencing, which I will refer to as mRNA-Seq today. mRNA-Seq can be thought of as sequencing all transcripts in the eukaryotic cell for qualitative and quantitative analysis. Using NGS, we can measure two things with eukaryotic mRNA-Seq. The first is whether a gene is active. We can measure the gene expression levels. The second is whether there are any changes in the protein that gene encodes. This may refer to a single nucleotide polymorphism, a SNP, or an insertion deletion, an indel, or even a structural variant or an alternative splice variant. Novogene worked with the Huazhong University of Science and Technology to find out, can mRNA-Seq help us to solve this neuroblastoma puzzle? Lee and colleagues have now published this neuroblastoma story, as you can see published in Nature Communications. The study material was human neuroblastoma cell lines. The essential background information: 15% of childhood cancer mortality is due to this neuroblastoma, which I will refer to also as NB. The prognosis varies wildly. Some children do not suffer greatly, and others have a very complicated disease course and do not survive. Armadillo, or ARM, family genes have been implicated in poor prognosis. So the goal of this process was to find out if mRNA-Seq could help identify which loci are interacting with these ARMs. These are the candidates for treatment. Candidate gene ARMC12 was picked by analyzing databases and patient data. It was then transfected into cells, and this guided mRNA-Seq. This is the experimental design. As you can see, there are two pathways. The first is the SHSY5Y cells, so these are the cell lines, and that had a transfection with ARMC12. The other are the empty vector, the mock, that serves as a control. Novogene performed the RNA-Seq. We participated also in differential expressed gene profiling, as well as helping out with overlapping analysis of ARMC12 correlated genes, although we did not participate in the mining of microarray datasets that these authors engaged in. Important to note is that these authors followed up their study with CHIP, chromatin immunoprecipitation, and quantitative PCR assays. These were used to explore the data further and to validate the findings from mRNA-Seq. The authors' results did support their hypothesis. ARMC12 expression affected prognosis, and its interaction partners were identified. I will show you a few figures to help understand part of what Novogene can help you achieve. In the figure on screen, we have a volcano plot. On the left, we see the log two-fold change of downregulation and the upregulation on the right. We see that 3,200 genes were downregulated and 2,900 genes were upregulated due to the presence or absence of ARMC12. So we saw a very large gene expression change. This helps us to understand that this gene is very important to cancer. We also saw that these upregulated genes correlated with clinical progression. The Venn diagram on the left shows the RNA-Seq data, as well as ChIP-seq data and other information that helps us to analyze together different data sets. We also have a heat map shown at right to compare between the mock and the ARMC12 transfected cell lines. And you can see some very different patterns among genes that have different roles in the cell. Finally, I do want to point out the qPCR experiments validated the mRNA-Seq. We learned from this study that extra ARMC12 actually may help other transcription factors to bind and affects the pathway that the cancer will take in the patient. So, why did this experiment work? Let's consider eukaryotic mRNA-Seq compared to other methods. First, as a review to make sure everyone is comfortable. When we study gene expression, we typically start from an entire organism. Many of our clients start with mice or rats. In this cartoon, I am showing you a mouse from which we have isolated tissue, in this case, the brain. From that tissue, we isolate RNA, and then we need to count the transcripts in some way. This is obviously a complicated process, and biologists have been working on this for many years. Right now, the gold standard is considered to be quantitative PCR, where you can amplify a target and compare to a known target to assess copy number. We also know that SNP typing can work and microarray typing can work. These often depend on hybridization or some sort of bait and capture. And in this case, the copy number is determined often by fluorescence. There's also a sequencing-based set of approaches in which we can count sequences to assess copy number. So when we actually do this for one of our experiments, notice we can compare an experiment and a control. So the experimental condition is going to be shown on top, the control on bottom. First, we isolate tissue, then we isolate RNA. If we are going to use eukaryotic mRNA-Seq, we're going to sequence the pool of all those RNAs that we extracted. Now, I do want to highlight that this technology can be applied to single cell. This is not the focus of today's webinar, but please contact tech support if you want to learn more. Most of our clients are interested in looking at pools of cells because they're interested in tumor dynamics or comparing tumor and normal or disease patterns that do not rely on single cell analysis. Once we have sequenced that pool of RNA, we can continue to analysis. I also want to highlight mRNA-Seq provides quantification as well as coding changes. Here, we again have the experimental and the control samples. You can see that gene 1 is downregulated in this experimental compared to the control. You can also see that gene 1 carries a novel mutation in the experimental compared to the control. My training is in population genetics and genomics, so I am very interested in these kinds of mutations. Maybe you are too. Overall, there are many ways that we can pursue understanding what's happening in the transcriptome. Novogene supports mRNA-Seq. We believe it is fast, inexpensive, quantitative, and can be used without knowing what is present before starting. Plus, we gain that sequence information that may help our projects. When you compare mRNA-Seq to Sanger sequencing, microarrays, SNP typing, or quantitative PCR, you see that it has many gains. We especially recommend NGS mRNA-Seq versus microarrays, and I want to take a moment to talk about this. In microarrays, the relative intensity is considered to represent the expression level. This can be hard to interpret if you do not have a good calibration system in place. Whereas in mRNA-Seq, the sequencing reads count as the expression level. Now, it is understood that maybe this is an estimate because we're looking at more than one cell at a time in both of these methods, but counting sequencing reads is more secure for many investigators than using relative intensity. Aside from this, however, we notice that microarrays sometimes have low sensitivity depending on the design. They may have a low dynamic range where we're limited in understanding the lowest of the low and the highest of the high expression. We really are limited to known transcripts, and we don't often succeed in achieving alternative splicing information. However, this is very inexpensive and often accessible to many labs. In contrast, mRNA sequencing has very high sensitivity. It has a high dynamic range. We can sequence a single molecule up to several thousand or million molecules. Novel transcripts can be identified, and we have the power to detect structural variants and alternative splicing more easily. We may also be able to do unlimited sample comparisons, even though the price is not quite as low as microarrays, although I will say that our prices, we hope, are competitive. I won't be talking extensively about pricing. I saw there was a question about this, because there is price difference between every country that we serve. And also this depends on the number of samples you have prepared and the current promotions. But we are working on bringing these prices down for our clients. And we encourage you to talk to sales about this. So one thing I would like to say is we will be meeting additional projects today from a couple of other clients as we proceed through this presentation. Take a moment now to reflect. What biological questions might finally be answerable with NGS-powered eukaryotic mRNA-Seq? Now I'd like to talk to you about the rest of the contents of this presentation. We have already discussed how eukaryotic mRNA-Seq helped us to understand neuroblastoma better. Next, we'll move on to details of mRNA-Seq and how this method can be used in agricultural projects. We'll then talk about how Novogene's workflow produces these data. We'll discuss an overview of our analysis options and how those analyses helped us to understand prostate cancer better. Finally, I hope to convince you why you should work with Novogene. We are experienced and we are dedicated to helping you do your very best science. So let's move on to the mRNA-Seq details. As most people in the audience today know, in eukaryotic mRNA-Seq, we tend to focus on the protein coding transcripts, but we definitely are working on capturing any transcripts bearing a 5' cap and a 3' poly A tail. The table at left shows us many different kinds of RNAs that are present in the eukaryotic cell. For instance, the coding RNA, the messenger RNA, which is our focus, but many non-coding RNAs, including linear long non-coding RNAs that may be regulatory in action. We also acknowledge that there are many housekeeping or baseline RNAs that are important for cellular function, including transfer RNAs and ribosomal RNAs. I'd like you to know that Novogene does offer long non-coding RNA, small RNA, and whole transcriptome sequencing. We can also support FFPE and ribodepletion if those are of interest to you, and we can talk more about that in the Q&A, but you can also email us to learn more about these options. Today, again, we'll focus on the basic mRNA process. So why do people use this process? Many common research reasons for using mRNA-Seq include comparing gene expression levels. Let's say you're working in a single species and you're looking at the same tissue in one or more samples. You may be able to differentiate between experimental treatments and controls or different experimental treatments, or you can look at different time points. This is important for many people who are studying developmental biology or understanding processes of aging. We may also look at different tissues where we look at gene expression differences, say between the liver and the kidneys, or between the brain and the heart. This may help us with understanding cancer biology, but also how metabolic processes may differ across an animal or a plant. In the same tissue and different species, we may be able to gain evolutionary insights. This may be true for understanding the history of breeding in an agricultural project, in a particular animal or plant, or it could be about general evolutionary biology. We also see many people studying gene function inference. We can understand cell-specific patterns, disease-related patterns, and treatment-related patterns, possibly used for disease diagnosis or prediction. We also see many people studying gene networks and pathways, where the gene expression levels help us to understand what function may belong to a gene and to understand links between genes that are interacting for cell metabolism or other purposes. Our clients have made these our most popular project types. Many of our clients are interested in medical research, even outside the clinic. We see people studying pathological mechanisms, for example, oncogenes or fusion gene analysis. Our clients investigate tumor subtypes and their classification, molecular markers for a variety of studies, drug targeting, in which case the gene expression variation can help us understand gene function, drug function, or even side effects. Some of our clients are studying human evolution for both clinical and basic research purposes. We do offer some clinical diagnostics as well at Novogene. And personal healthcare is a growing interest for many clients. Here, we can study disease-related gene analysis, including oncogenes and fusion genes again. However, many of our clients also come from agriculture. This may be for development or adaptability, for crop evolution, or for understanding agronomic traits. In particular, gene expression and functional analysis between ancestors and offspring can be very interesting. Please understand that we can accommodate a wide variety of eukaryotes here at Novogene. If you don't see your system represented, we may still be able to help you, and we encourage you to contact us for more information. Let's turn now to that agricultural case. Agricultural scientists often need to understand the history and function of modern cultivars. Let's discuss wheat. Modern hexaploid wheat is a hybrid of a 4N and a 2N species. On the left, we have an image of Triticum turgidum, which is represented by the AABB genome type. On the right, we have Aegilops toshii, which is represented by the DD genome type. We have understood that these two species likely formed a hexaploid represented by the AABBDD genome type. This allohexaploid is considered to be what we have as modern wheat that most of us encounter in our meals. This modern hexaploid outperforms its parents. This is important for modern breeding studies and also for understanding the nutritional needs of a changing world. Novogene was able to help Lee et al. Study their novel hybrids and the offspring of those hybrids using mRNA-Seq and, interestingly, small RNA-Seq. Let's explore. Ai-Li Lee and colleagues have published this wheat history story in the plant cell. Their study material was the Triticum turgidum genome, Aegilops toshii, the hybrid offspring, and their interbred offspring. The essential background I want to share with you is that modern wheat, Triticum aestivum, started as this hybrid and has been developed over time. We know that hybrids outperform parents, but the basis of this is unknown. We don't know if this is primarily due to coding changes, expression changes, a combination of both, or something more related to homolog control. As a reminder, homologs are homologs found in the same genome due to the increase in copy number of a gene through mechanisms like genome duplication. The goal of this study was to use mRNA-Seq and small RNA-Seq to identify which loci affected this hybrid offspring success to understand better what's happening here. I would like to show you that seedlings of many samples were analyzed using RNA-Seq. The breeding and sampling scheme is shown at left, where we have the parental species that were examined, as well as the primary hybrid and the interbred offspring. The RNA sequencing scheme, the experimental design, is shown at right: 7-day-old seedlings, spikes at the heading date, immature seeds. What I would like to highlight is that Novogene again helped with RNA-Seq, both for mRNA and small RNA. We helped with the differential expression analysis of both. We do have the ability to analyze microRNAs in some cases, and we provide annotations for gene ontology and other annotation types, which we'll talk about more later. As in our last study, we noticed that CHIP and qPCR assays were explored and validated. What became fascinating was that 12 expression patterns were found among all these different organisms, and candidate pathways became clear. At the top of the slide, we have a table indicating these unique patterns of gene differential expression compared to the parental type, the ELD. We also see at the bottom that genes involved in catalytic activity, response to stimuli, even the cell membrane, were involved in this differential gene expression and may point to ways in which the offspring differ from the parents. Using both mRNA and small RNA provided additional insights into breeding history and biology. On the left, we have figures from this paper that are showing that non-additive expression of microRNAs was important to growth vigor and adaptation. On the right, we see that small interfering RNAs act as a genetic buffer and help to regulate homolog expression. Let's take a moment to reflect. What types of RNA are important to your study system? And could using more of Novogene's RNA sequencing services provide greater depth to your experiment? I'd also like to take a moment to answer a couple of the questions that are coming in. One of our guests asks, which verification methods are used nowadays? We're still using qPCR for many of these. However, some people find, depending on your journal or organization, your professional society, that other types of experiments are able to validate these data. Generally, our clients are interested in using RT-PCR. I'd also like to answer a question about whether private non-academic companies are able to use our technologies. Absolutely, we have many clients from at least across the United States that I have worked with personally who have maybe limited income but are doing development of how to do libraries. We can work with you as well. There's a question about bacterial changes. We plan to have another webinar later to answer our prokaryotic systems. Okay, let's return to the planned webinar now. The next section will be about how Novogene's workflow produces these kinds of data. Here is our basic workflow. We start with total RNA. You send us tubes of samples. We then do sample quality control. We prepare the libraries. We do an internal library quality control assessment. We move to sequencing, and we do data quality control. In the next section, we'll discuss more deeply the bioinformatic analyses that are options for our clients. But before I talk about all of this work, I want to highlight an interesting fact. Our network of offices and labs is global, and some of our teams are specialized. What you see on this map in blue are countries and regions where Novogene has successfully conducted projects with our clients. And here are all of our research facilities. Especially during this pandemic time, we may need to transfer your sample to another lab in order to assure the very highest quality and the fastest turnaround time. Please talk to your technical support scientists if you have questions about this process as a client of Novogene. Today, I am very proud to represent Novogene Americas. We manage all of the business from North and South America. Our lab facility is located at the UC Davis Medical Center, and we have a really wonderful team standing by to answer all of your needs. Again, this is a pandemic time. California has us working from home, but we are in touch with our clients all of the time, and we are here to answer your questions. As you can see, though, we have many lab facilities around the world, and we deal with many different kinds of samples. Let's focus for a moment on the sample types that we accept. I'll highlight for today total RNA obtained from fresh tissue. We also accept total RNA from SNAP frozen tissue. This is usually very high quality, in fact. Our required minimum amount is 0.4 micrograms. This is the same as 400 nanograms. This is the minimum amount that we require, for which we know, based on the repetitive work that we do, that we can guarantee that your library should have usable, reliable results. If you have less than this amount, stay tuned. We are planning a webinar on low input eukaryotic mRNA-Seq for the future, or contact your sales or technical support to help you with understanding these other product types. In general, we ask for at least 20 microliters of sample. This helps the samples survive shipping. It also allows us to have enough for our quality control as well as the experiment. Ideally, we're looking for an RNA integrity number of greater than 6.8 for animals and greater than 6.3 for plants or fungi. And we understand that insects have unusual readings as well. There is variation possible here, but we like to say that this is where we know our libraries will be good. We do ask that you achieve a concentration of 20 nanograms per microliter if possible. This helps ensure a more reliable outcome from the sample QC process. In general, we're looking for intact mRNA. We're looking for that to be clean of any contaminants. As you can see here, we have total RNA isolated from blood we can accept. We have guidelines on fresh tissue from animals and plants, numbers of cells, even blood samples. So there's a question coming in about whether we can actually do extractions. We can help with many extraction types, yes. And I want to highlight that the closer you are to meeting the standards that we have set, the better we're able to guarantee the data output quality and quantity. But please do inquire for your special cases. Our primary means of sample quality control is using the Agilent bioanalyzer system. Here in UC Davis in Sacramento, we have the Agilent 2100 system right now. At some of our labs, we also have the Agilent 5400. The first thing we look at is the reconstructed gel electrophoresis. This is an easy visual way to understand whether there was any degradation. We also can ask whether the quantity is sufficient for library preparation. We're also looking at the RIN and the estimate of the concentration of RNA in the sample to ask, was there any degradation? Is there any contamination? Do we have enough material to start? Shown here are some examples of what happens when we use this system. For library construction, I won't go into extensive detail, but I do want to highlight that we use trusted kits from Illumina and New England Biolabs. We take the input mRNA, we ensure that the pieces are the right size for the downstream Illumina platform that we favor. We then do the necessary chemical reactions to attach the adapters necessary for the Illumina system. Fairly straightforward. I also want to highlight that here in UC Davis, we do have automation with robots to help reduce human error and contamination. This does also normally speed up our projects, but it may limit our ability to do custom projects as quickly as you may need. Please consult us for further information. I am also very excited today to tell you that we have a new intelligent platform called Falcon. We are adding these to help us provide even faster, more reliable data. Notice that the Falcon platform provides you with a whole suite of robots. I look at these pictures and I am so fascinated. We have a series of robots that can automate everything from sample extraction to bioinformatic analysis. This may help us process as many as 2,000 to 3,000 samples per 24 hours and may reduce our turnaround time by 60%. When we release these slides to you after the webinar, we will provide a link to a video where you can watch more about the Falcon system. I've mentioned Illumina a couple of times now. I do want to acknowledge that there are many kinds of sequencing platforms available. Illumina is just one of them. We favor Illumina, however, and we are an Illumina certified service provider. I've provided a schematic of their workflow here. One thing that I have not discussed in detail is our internal library quality control process. When we are doing this work, we are looking for the sizing being correct, the concentration being correct, the molarity being correct, all guaranteed to help us make sure that our Illumina runs are working correctly. The advantages of Illumina are its high throughput, its good accuracy, the fact that it is well tested, and the fact that it is community trusted. This makes Illumina our choice for eukaryotic mRNA-Seq. Let me tell you a little bit about one of our machines. This is the Novogene 6000. This is my favorite Illumina machine. Right now, we primarily use the S4 flow cell. The output per flow cell can be approximately 3 gigabases of sequence. That's 3 terabases of sequence. And there are 2 of these per run. So we're looking at 6 terabases of sequence data per approximate 44-hour runtime. We are very proud to have these around the world in all of our labs. We offer a very high quality score promise, where the Q30 is going to be greater than 80% of the bases we sequence for you. We can guarantee this for you if samples pass our quality control test. And I personally have seen numbers closer to 95% for many client projects. The sequencing strategy we use is PE150. That means 150 bases are sequenced from each side of every fragment that is captured in library preparation. And we recommend a data amount of 20 million raw read pairs per sample. I'd like to emphasize this means that you get 20 million pairs of sequence reads. And this may actually help to understand our services compared to some competitors who offer 20 million reads, but it's only 10 million pairs. Data quality control comes next. The main things we look at first are error rate distribution, GC content distribution, which can be perturbed if our reagents are not perfect or if something goes wrong during sequencing, and the composition of raw data. We have a triple data filtration system. We filter for reads containing too many uncertain base calls. We filter for too many low quality base calls. And we filter for any reads that appear to have some or all of the adapter, which may indicate a problem during library synthesis or something unusual about the project. I'd like to highlight that data quality control is always free in the projects that we offer. Let's take a moment to reflect. Can Novogene help you optimize your samples for library preparation? What custom options would you like to ask our technical support about? We have a question from one of our attendees about how well this method can work with flash frozen mouse brain. Brain is a little more delicate than some tissues. We actually have a neuroscientist on staff who would be happy to talk more with you about this. But in general, flash frozen mouse tissues have had good success with us. There's a question about analysis that I'll get to in a moment. We also have a question about examples of analyzing metabolism. And I'm going to show another example that I hope answers this, but please follow up if it's not sufficient. Let's return to our slides. Now that I've talked about neuroblastoma investigation, agricultural investigation of wheat, and how we produce our data, let's talk about an overview of our analysis options and how those analyses can help us to understand prostate cancer better. First of all, I want to note we offer many bioinformatic analysis options. This is not a surprise. We were founded by the biologist who wrote SOAP and SOAP de novo, Dr. Ri-Chung Lee. My apologies, something went wrong with the title here. For eukaryotic mRNA-Seq, we offer data quality control packages, quantification packages, and standard packages. Let me walk you through these options. In the first case, data quality control, we ask, did the sequencing itself work? You can ask us about delivery of clean reads. We can provide these triple filtered reads to you as a separate data set in addition to your raw data. This is free if it's included in your initial quote. We also offer a quantification analysis package. This provides gene expression levels and some of the related analyses, plus all of the data quality control. Finally, we offer what we call our standard package. This includes mutations and other differences in structure, as well as data quality control and quantification data. Here is our quantification package, and again, my apologies for the title. This may look complicated to many clients, but sales and technical support are standing by to help you pick the best analysis package for your science. And one of our panelists today who can answer questions in real time is from our bioinformatics department. Quickly, I want to highlight that we have raw data that gets filtered and analyzed during data QC. When we have a reference genome, we then can do gene expression quantification, analysis of the differentially expressed genes, and we pursue functional analysis after that. Enrichment analysis and protein-protein interaction are available for all eukaryotic systems. We are able to add oncogene analysis and reactome enrichment if you have human or mouse samples, and disease ontology and dysgenet enrichment if you have human samples. This only shows you the actual analyses. We provide multiple figures and tables for many of these to you. Let's talk about another client paper. Kim and colleagues have now published this prostate cancer story. Their study material was the 22RV1 human prostate cancer cell line. The essential background here is that withafferin A, WA, may have an anti-cancer property, and preliminary data suggest that WA acts on metabolic pathways, but its specific mechanism of action remained unknown, and the authors wanted to find out what specific pathways were affected. Their goal was to use mRNA-Seq to identify which pathways are affected by WA. Here is a schematic of their experiment. Cell lines both with and without WA were sequenced using RNA-Seq in triplicate to ensure statistical analysis could be done. And notice that Novogene provided differential gene expression analysis as well as gene functional enrichment. I'm going to show you a series of figures and talk about what they mean in general. Here at the top, we see a summary of RNA-Seq mapping results, which Novogene provides as an Excel table. On the left, we also see a volcano diagram, a Venn diagram, and a heat map, all of which are offered as publication-ready figures by Novogene. And what you can see is that the treatment, the differential use of WA, did have a huge impact on the transcriptome. We also offer these kinds of figures for GO and KEG. When you have a human sample, the DO and reactome are also available. These are figures that show enrichment by pathway. So MAPK signaling, cancer pathways, autophagy, mitochondrial intermembrane, translation, and cellular response to external stimuli all showed enrichment in one part of this experiment. As you can see, every row here is a different category within that particular database analysis type. So KEGG has one type of analysis, gene ontology has another. Please feel free to request a demonstration report from us to see detailed figures and tables, including what software we use and the major parameters we employ. Let's take a moment to reflect. How can Novogene's analysis packages help you analyze your data for today's questions and for tomorrow's questions? Our final section is where I hope to talk to you about why this company is a good choice for you. Novogene offers high-quality sequencing and analysis, but so do many other companies. Why should you pick Novogene? Our core values are client-centered. Our goal is to help your project succeed. Our driving mission is to be the global leader in providing genomic services and solutions. We pride ourselves on our professionalism, our innovation, our partnership, and our integrity. We have the experience to achieve our high expectations for your project, even during this time of pandemic. We have processed thousands of RNA samples, from fresh to museum or FFPE samples. Many of the Novogene USA staff hold advanced degrees in science, so we understand what it means to write grants, publish findings, and present at conferences. We do our best to help you be a successful scientist. I am incredibly proud to help run the technical support team and want to quickly introduce to you Oliver, Manoj, and Mario, who are answering today's questions on the panel. We are also proud to say that we have many partners across research types and across the world. Over 2,600 research organizations and universities, over 1,200 companies, including small biotechs in food testing, agriculture, and other areas. We also have partnered with over 590 hospitals, and we hope that we can partner with you. I want to take a moment to thank our clients who generously allowed us to share their publications with you today, and I encourage you to read their papers after the webinar. I also want to thank you for your attention. There are many ways to contact Novogene. Try our website where queries can be submitted. Email us to be connected with someone who can speak your language. Follow us on social media or connect with us there to learn more about what we do and see our time-related promotions. I hope you enjoyed this webinar. Our series continues next week, and we would love to know from you in your survey results if you would like to see an advanced eukaryotic mRNA-Seq. Please, I'm seeing questions about viruses and bacteria. We would love to talk to you about those in a separate webinar as well. Let us know. And follow us on LinkedIn. This is where you can find a lot of our webinar announcements. Thank you for your attention today, and thank you for so many wonderful questions. Now I get to spend some time answering them. I will remind you to please use Zoom's Q&A tool to help ask these questions. And I want to note, if we do not get to your question here, please submit it on Zoom. Zoom will catalog these, and we will try to answer you directly by email in the next week or two. Okay, I'm going to take a moment to check to see which questions have come in. I think most of them, we've talked already about whether we can help with researchers versus private companies. We can. I even know of someone who was helping his father with a personal medical condition, and he privately contacted us for some help with his father's medical team. We do definitely help people in almost every sector. We do not require that you be a member of any sector to do business with us. I also want to highlight that if any of this has gone fast or you want to revisit it, we are going to be sending you some slides later, and the recording will be made available. So one of the questions that we have, again, about viruses, we do have the ability to help with certain kinds of projects, but this is the kind of question we want to have you talk to technical support about, because we have to examine biosafety to protect our coworkers, but we're happy to talk to you about options we have for this. There's a question about quorum sensing. We can talk to you more about this when we talk about prokaryotic mRNA-Seq. We do offer this as a separate product type. There's a question about price. So I'll give you some of our estimates. I know that we do have a variety of promotions on now. Some of them can be as low as about $175 per sample. And if you have the right number of samples, that may include some free analysis at the quantification level. Again, the price varies based on the number of samples that you submit to us. If you have a large number of samples, like 1,000 samples, we actually have permission to ask for a special discount for you. I am really glad to see that one of our comments is telling us that we are joined by people from all over the world today. Thank you for joining us from Iraq. And here is a question that I am very excited to answer. What strategy can we use for species with an unavailable reference genome? If you'll bear with me for a moment here. We do have a de novo transcriptome analysis package. So we start with the same data quality control. And instead of mapping to a reference genome, we use the software Trinity right now as the anchor point for assembling our reads into a transcriptome assembly. We can then map the reads back to that assembly to count how many line up with the different parts of the transcript, of the transcriptome. And we use multiple databases to help provide potential annotations here. So once we have that transcript assembly, we can do SSR analysis, SNP and indel analysis. We can also do gene functional annotation. So you can see we do GO, COG, and KEG. We can predict coding domain sequences, and we can do a quantification and differential expression analysis. And we can provide some functional analysis of the genes that we see that appear to show this differential expression. We can sometimes offer additional specialization, and our bioinformatics team is constantly updating our packages, so please inquire for the most recent information. Okay, here's a great question. After RNA-seq analysis of your tissues, in this case, a client studying pigs, do you have to conduct real-time qPCR? Novogene does not tell you what you have to do. We are here to support your experiment. But my experience being a lab researcher is that my first place to go is to anyone providing the grant money or the people who will be reviewing the paper that I need to submit. Oftentimes, societies will have standards. So I've been a member of the Society for Molecular Biology and Evolution here in the US. They actually have standards for what needs to be validated during your experiment using different methods. So I would say it depends on your experimental goals and whether you're in a situation where you're publishing for the public or if you're using it for in-house material. My suspicion is that after a number of additional years, Illumina has become so trusted, we may not actually see as many people needing to have this extensive validation, but qPCR can reassure many of our fellow scientists. I have a question about ribosomal RNA depletion. Right now, I think we still have enough from Illumina that we're using those. The lab was investigating because Illumina started selling their ribodepletion kits in a different way. We are in the middle of testing those to see which are the optimal, but because we are a major client of Illumina, we were able to buy a lot from them. There's a question about whether I think or we think that mRNA-Seq data should be validated. And I think this depends on the goal of your project. Certainly, if you're interested in drug discovery, if you're interested in setting up a clinical test, I'm a little bit more conservative in my science. I do want to see multiple lines of evidence saying that this is the right way to go because I don't want to see a living thing hurt by a wrong answer that we may have gotten accidentally. However, as a way to find candidate genes, which is something that has been true in some of my research, I think that mRNA-Seq is a fantastic method. And I think that many experiments, if you achieve the right coverage, don't rely as heavily on needing that validation. We're happy to talk to you. My team, I am so proud of my team. My team is excellent in the technical support department. They can help you understand whether this validation is going to be super important for your project. But again, we don't want to tell you what to do. You know your science the best. We're here to help make sure that you're successful. Oh, here's a question for us about what differentiates us from our closest competitors. One thing I'd like to say is, for me, I think that my closest competitor is myself. I always want to do a better job for our clients. I always want to step up what I am doing for my clients. However, we do actually pay attention to what our competitors are doing. Some of them have much longer turnaround times. Some of them have higher prices. Many of them we've noticed when our clients have compared with us or asked for price matching, which we are able to do often, they may not be offering the same amount of data or as much analysis as we're able to offer at the same time. But for me, the customer service is one of the biggest things that Novogene offers. We are here to help make sure that your science is good. And we have assembled a really excellent team with background in forensics, in medicine, in agricultural sciences, in neuroscience, people who have worked on microbiomes, people who have worked on viruses. We have a really diverse team. And I think that that's one of our major strengths and sets us ahead of some of our competitors. Let's see. I do want to take a moment. There's a question about coronavirus. We do not have coronavirus kits available for all regions right now due to biosafety concerns and local regulations. But if you have concerns about detecting coronavirus, please feel free to reach out. We do have a department that's working on this right now. Obviously, this is on everyone's mind, as all scientists around the world are either part of the process to try and solve it or being affected by this disease. So again, please inquire for the specifics of your case. And the question about the 175 per sample, if you meet the right requirements for number of samples, this can in fact pay for the sequencing and the analysis. But the packages keep dropping in price, so talk to sales to find out how low our prices can go for you right now. There's a question that I want to definitely answer. Can we share the presentation in video? Absolutely. This is coming to you as soon as we're done today. You submitted your email address as part of your registration, and so this is one way that we'll make this available to you. And we plan to make all of our series available to you. So next week when we talk about microbial amplicon and biodiversity sequencing, that will be available. When we talk about whole exome sequencing, that will also be made available. Here's a great question. Do we offer whole genome sequencing? Absolutely, we do. We have a special pipeline just for humans. We also have one for plants and animals. We have multiple pipelines for microbial organisms, some for fungi, some for bacteria. If you have a protist that you're working with, just call us, email us, let us know what you're interested in. This is another question close to my heart. The guest asks, can RNA-Seq analysis help determine the genetic structure of different populations of a pathogen together with genome analysis? We do have the ability to help do sequencing of the genome as well as the RNA present in those cells. The analysis of both together would be considered a custom analysis, and so that's something that we'd want to have you consult with our bioinformatics team about to see what tools we already have in place for that. In terms of population structure, I know we've just added some population genetic analysis, and I'm super excited about that. That was partly because I am so excited about it. Here is a really good question. Can we do online explanations? Can we work with you online? You are very far from our offices? Absolutely. We have offices in the United States, in California, and in North Carolina. So we have 2 different time zones for Novogene USA. But we have sister divisions that are serving Africa, the Middle East, India, Australia, Southeast Asia, and of course, headquarters in China. We have a new set of companies in the EU and in Great Britain. We also have some representation. We have a new company in Japan. So we'll help you find the right group that is closest to you, but we are happy to work with you by email, by phone, by Zoom, or GoToMeeting, or any other platform that's available to us. All right, we have a great question here. I can't check the RNA myself, but I checked the quality through gel electrophoresis and it was okay. Is this enough? Absolutely. Most of our clients do not have access to something like the bioanalyzer. It is very expensive. We only ask that you give us some approximation because occasionally during shipment, something can happen to your sample and we want to verify that it arrives in the condition you sent it to us. Any kind of information you have, if it's gel electrophoresis, if it's using the nanodrop, we can work with you. We encourage you to send this to your technical support so they can help analyze it and see if there are any red flags, any danger signs, and tell you whether the samples are ready to send. I also want to say here in the middle of California, it's getting very hot. We're already at 42 degrees today for our daytime high. So we do encourage you to consider RNA stabilizers or a lot of dry ice so that your samples get here intact. The turnaround time is another question we have for 10 samples, for example. Right now, our turnaround time is a little bit modified due to coronavirus, and I would say please submit your question. It also depends on whether you have special needs. For instance, if we find contamination that needs to be cleaned up, it may be extended. It's going to be at least a couple weeks at the very, very fastest that we're able to achieve, but we'll give you a specific example when you contact sales or technical support. Wonderful question about statistics. Do you always need at least 3 biological replicates, Or could you pool them or do samples in another way? This is a statistical question. This is best discussed with the people who are doing your analysis. My training in population genetics says that the more replicates we have, the better, that 3 is the absolute smallest number to make sure that we don't have an outlier captured in our data. But this is something that we're also happy to talk to you about. Many of our clients can adjust the coverage amount versus the total data amount, and we're able to help. All right, let's see. I think I have 1 or 2 more questions here before we run out of time. One, can we do genome sequencing of fungi? We absolutely can. There's a question about the RNA-Seq analyses. Let me send you a demonstration report so that you can see every single one. We have a couple dozen that we use. And here's a wonderful question. Can we accept samples shipped that are RNA from countries in South America? Yes, we do this every week. We can help you with this. I definitely want to give a special thanks to our main salesperson, Diego Levi, who has been leading our Latin American team. He is based in Argentina, and he can help you with any shipping needs that you might have, as well as our fabulous technical support team. Daphne, Oliver, Mario, and a few others are helping out. We also have staff who speak Spanish and Portuguese who can help you prepare your samples and ship them from Latin America. So we are in a position to help Latin American clients. Even if dry ice is not available, we do have some other recommendations that we can give to you. Let me see if there are any last questions that I can squeeze in. It looks like these are the most. The sequencing depth, absolutely. This is a good question. How do we determine sequencing depth? We actually are looking at the amount of coverage. So we can, when we look at the mapping data, we look at how many reads can map to a certain area. We look at distribution across the entire genome. Obviously, some disease pathways will modify this, but we're looking at the amount of gigs of data and actually the fold coverage as well. We can get you much more information about that from the bioinformatics team. Okay. I would like to thank also Sibi, who is our organizer today. He is the supervisor for the marketing team here in the United States. Sibi, are there any other things that you'd like to remind our guests about before we close up shop today?
Yes, we just wanted to remind everyone and thank that we will be sending out a follow-up email with the slides and the recording of the webinar. Thank you for joining us today. We appreciate you guys joining us. If you have any questions, please feel free to let us know through our contact us page on Novogene's website at en.novogene.com or by sending us an email at inquiry_us@novogene.com. You can also stay connected with us and engage with us on social media. We are very responsive and post updates on promotions, future webinars, and other important information. Thank you again. Have a great rest of your day. We look forward to working with you. Take care. Bye-bye.